This week, I had the opportunity to connect with Dr. Stephen J., who is an associate professor in the Fishkill Department of Bioengineering at the University of Maryland. Stephen, who previously did his postdoc in Rich Lee's lab at the Harvard Medical School and was a visiting scientist at MIT, has a deep passion in drug delivery and molecular biology. In this interview, we were able to gain some insights as to how his work in extracellular vesicles have evolved and his perspective on its future. Good morning, Stephen. So it's great to have you here with us today. So I wanted to kind of take this opportunity to chat with you and learn more about some of the work that you're doing. So I know that your lab is focused mostly on the design and development of novel therapeutics, especially extracellular vesicles and exosomes. And this also sort of overlaps with some of your interests in tissue engineering. I wanted to start off by talking a little bit about the extracellular vesicles and exosome work that you're doing. How did you get into this research, and what made you sort of interested in this field?
Sure. Well, first off, thanks for having me on and talking to me. How I got into it is sort of a long story, so I'll try to shorten it. So I was doing a PhD with Mark Saltzman in polymer-based drug delivery. And I got exposure to molecular biology through a collaboration with Jordan Pover's lab at Yale during that time. And I became really interested in that and wanted to learn more about it, but still had this sort of therapeutic aspect from drug delivery in mind. So I joined Rich Lee's lab at Harvard looking at applying molecular biology to therapeutics. And while I was working there, I got to work with a medical resident named Jason Rowe, and he's actually the one who was doing a small independent project related to exosomes, as they were referred to at the time, and that kind of opened my eyes to the emerging field of EVs. And this was around 2011, 2012. So some of the really seminal work out of like Matt Wood's group in Oxford and stuff was just coming out at that time. And so that kind of got my attention. And I realized that this was something that combined my, interest and expertise in drug delivery with my interest in molecular biology, and it was just sort of taking off. So there was some space to work and operate there as I was looking for a faculty position and starting the lab. So it just sort of all came together. And, so I decided to go for it and made it a feature of my new lab, even though I didn't really have any background. I had no publications with, exosomes or EVs or anything when I became a faculty member. So, really a lot of hard work by some folks in my group, Tekla Machane, Divya Patel, Anjana Jayaram, that really made that happen and allowed us to actually become established in the field.
That's awesome. Yeah, that's really exciting. I think the field of EV has really evolved very quickly over the last 10 years. So I think it's great that you were right there, when this all started. So I think that's really exciting opportunity for all of us. You mentioned drug delivery. I know you did a lot of work using extracellular vesicles as a vehicle for drug delivery. What have you found to be sort of most challenging working with EVs, I guess, as a carrier?
Well, I mean, the most challenging part is they're also the greatest asset, is their biological in origin. And so, it gives you, on the plus side, you can apply all the power of molecular biology, genetic engineering to designing, a therapeutic that's, in an EV. But you also have all the things that come along with that. So there's always going to be biological components. It's going to be almost impossible to completely characterize every single part of what's in an EV. How do you do quality control in terms of, the cells they're producing EVs are dynamic. And so how do you ensure that, batch 1 and batch 1,000 and batch 10,000 are the same? Sure. It's difficult. And how do you assess toxicity and potential off-target effects? These are going to be the biggest challenges, I think, going forward.
Yeah, definitely. I think the batch-to-batch variability is something that is, inherent with any biologics. So, that's definitely something that we're also always in discussions with a lot of the folks that we're working with in the field. So yeah, that's a very great point. So I saw one of your papers. You talked about, using EVs and loading a functional microRNA and utilizing this pH gradient modification. Can you share a little bit about sort of your thought process and how you came up with this idea?
Well, I mean, it's actually quite an old idea, and that was applied to liposomes many years ago. And so, like many of the, initial efforts that people have made for loading drugs into EVs,
Mm,
is kind of, see what's been done with artificial vesicles and try to apply the relevant things here. And so, yeah, rotating student in the lab, Ishan Dahal, he was one who came up with the idea to try this, that, we could try this technique from liposomes, and we had to adapt it somewhat. And, some other folks, Tekla Manchane, Anjana Jairam, really like, help complete that work and bring it to fruition,
mm,
along with a collaborator from the University of Maryland, Baltimore, Dr. Wei Chow. So, but so we had to do some optimization and some minor innovations there, but ultimately, um, the idea was out there, and we just kind of
mm.
applied it
Sure.
to EVs.
Right. Sort of adapting something that has already been done in the liposome. So, and it worked really well with the EVs as well, I see.
Yep.
Awesome, awesome. Yeah, we also, always talk about, I think in this extracellular field, how challenging it is to get enough materials, right? Extracellular vesicles. So biomanufacturing is always something that we often discuss. I saw that you had a paper as well that actually used 3D printed scaffolds in sort of a perfusion bioreactor to show that you could actually get far better yields than in a 2D culture. So, what are some of the aspects about this process or system that you think made it unique in terms of its role in increasing the yield or even potency of the EVs?
Sure. Yeah. So this is a project we were, working on in collaboration with John Fisher at University of Maryland, who's an expert and developed this 3D printed perfusion bioreactor technology, and Divya Patel in our group really drove this work. And basically, I think there's a couple key components that contribute to the effects that we've seen. And the first is just the dynamic nature of the culture. So, when you have sort of maybe a conventional way to produce EVs is just grow cells in a flask under static conditions and allow the vesicles to be secreted out into the media. Then you collect the media at some certain time. But along with secretion, you're also having uptake by that same population of cells.
Mm,
Vesicles go up, they float around the media, but they'll get taken up as well by the cells. So if you just have media constantly flowing, then that uptake component is removed, and so
mm,
just by that nature, you get increased yields
mm,
by removing uptake from the producing cells. And then the second aspect is whether there's any real mechanobiological effects contributing to both production and bioactivity of vesicles in dynamic culture. So we know cells like MSCs are highly responsive to mechanical stimuli and
mm,
can potentially differentiate based on their substrate, the mechanical stiffness of their substrate, or the shear that they experience through the
mhm,
flow of media in a bioreactor. And so no one really knows very much about how those forces um, dictate outcomes with
mm.
respect to EVs.
Right.
So that's something that we're also studying quite earnestly in our lab. And, we suspect we have some initial indications that there are significant effects, but it still remains to be seen exactly what's
Mm.
going on there.
Right. Yeah, I think that's a sort of interesting question to answer as well, as far as mechanobiology and how that might affect the secretion of EVs or even characteristics of the different EVs that might be secreted due to those factors. So I thought that was a very interesting and unique approach. So I think it will be great to see a little bit more and understand a little bit more about how it's affecting the cells, the MSCs for sure. Yeah. So I'm always curious about learning everybody's past. So what led you to academia? What do you enjoy most about it, or what do you dislike about it so far in academic life?
Um, my father was a researcher, and so I've always kind of been into science from an early age.
Sure.
Um, and I had sort of different career interests at different points of growing up, medicine, marine biology,
Mm,
but I kind of always gravitated back towards research. And so that's kind of what led me on the path that I took. But it's a pretty conventional path. I mean, I went to college, I went to graduate school. I was lucky to have some, outstanding mentors
mm,
um, in my graduate work and my postdoctoral work, and then some great colleagues since I've joined the University of Maryland. And then I also in high school and college, became inspired by some teachers that I had, and saw
mm.
the impact that really great teachers can have um, on people's lives. And so, that also was a key part of it, was being able to impact people through teaching,
Right.
and also to be, both teaching and research allow you to be really a lifelong learner, right? I mean, you got
Yes.
to constantly be learning new things, and I enjoy that. So it's nice that, to have a job that actually, requires that, instead of that
Yeah.
you just being like what you do in your spare time.
Yeah.
So I appreciate that. I enjoy the sort of freedom, the
Mhm.
intellectual freedom.
Right.
I don't know what we're going to be doing in the lab in 5 or 10 years. Might be similar, might be totally different.
Sure.
But that's excites me. That doesn't, I think that's awesome. Instead of just kind of having the sense of, yeah, I'm in this job and I see what the
Sure.
next 30 years are going to be, and I'll just kind of turn the crank. In this case, I really don't know. It could be totally different. And I don't think there's many jobs that afford that opportunity.
Yeah.
Um, so that drew me in as well.
Awesome. Yeah, that's definitely right on point. I think the intellectual freedom in academic life is what's challenging, but also absolutely exciting. And I can totally relate to that. Absolutely. Yeah.
Yeah.
So, do you have any advice for new faculty members, or people who might be thinking about building their academic career, or some of the things that you've learned, or maybe mistakes in the past that other people could avoid?
Well, my advice is sort of paradoxical, because I would say that everyone's um, path is so unique that
Right.
my advice would be, don't pay too much attention to the advice that you get from other people. So that's sort of contradictory in terms, but it's true. I think that you know it's best to just sort of focus on setting your own goals, and don't worry too much about you know
Mm,
what people in your field or what people in your department are doing in turn. I mean, of course you keep up with the science and whatnot, but
yeah.
don't worry too much. Oh, this person got this grant or published that paper, and just kind of what you're trying to do, and try to find satisfaction in like reaching your own goals, right? Because there's
Right.
always, in science and academia, there's a lot of rejection, right? There's a
Yeah.
lot of negativity in everything you do. Even if you're doing really well, your papers are getting rejected, your grants are getting rejected at a very high rate.
Yeah.
And so sometimes people don't really understand what that's like. It's like, oh yeah, 80, 90% of the stuff you do gets rejected. And yeah, okay, and so that's some stretches, it's 100% for a while, and
Yeah.
you just have to kind of deal with that. So that's why I say, like, yeah, just set your own goals. You have the things that you're trying to do, and try to find satisfaction in the successes that you have, and then always keep in mind all the different areas you can have an impact, right? It's not
Right.
just publishing the big paper or getting the big grant. I mean, you're teaching, you're mentoring, you're doing outreach, and all those things are great. And again, most jobs like don't give you necessarily the opportunity to make impact in those
Mm,
areas. And so don't forget all these other things that you're doing that are making positive contributions, even if you know some of the other stuff you're focusing on, you're getting you know a lot of negative feedback for
yeah.
long periods of time.
Absolutely. I think that's great advice, because that's definitely in my journey too. Yeah, I think it's easy to get down with all the rejections from the grants and whatnot, but you just have to keep at it, right? And yeah, it's so true. I think the students always inspire me as well. When I was a faculty in NTU, so working with them and just hearing all these ideas they have and seeing them graduate, yeah, that's my source of inspiration too. So yeah, absolutely. Yeah, so we recently had this ISEV virtual conference. I wanted to kind of hear your thoughts about, what have you seen, what have progress, right, over the years in this field? What have you observed? And where do you think are some of the challenges for the field as a whole? I know you touched a little bit on that earlier, but can you maybe give sort of a high level overview of what are the next steps we need to take?
Sure. I mean, there's tons of things to be done. It's a pretty
Yeah.
young field overall, and there's issues related to delivery and biomanufacturing, as well as just understanding more about physiological roles of EVs to help impact therapeutic design and so forth. Um, I guess one of the encouraging developments for the last few years is there's been a movement within the field to kind of develop some standards and
Mm,
improved rigor in the research and improving reproducibility um, and that's so important, to making anything useful going forward. That you can understand better what everybody is doing around the world in the field, and be able to reproduce the key findings and to be able to build on things instead of just kind of everybody has their own little special batch and their special conditions, and you know can only make progress lab by lab instead of
right,
the whole field. So
right.
I think that's great, and I hope that'll continue to progress. um, I mentioned before about the quality control issue, and I think that's also a big thing, and maybe something that hasn't been um, thought of as much, or at least discussed as much. That deserves more, um, more thought in the future.
Mm,
There's certainly been focus on downstream quality control, but I think
mm,
upstream it has been really neglected. And thinking about ways to understand if we're running a bioreactor batch, and is there a way to understand if you know this is going to be a good batch or not before we go through all the trouble of processing, purifying the product, and then at the end we run some tests and figure out, oh, it's not good. Well,
yeah,
that's super costly, and it just diminishes the potential for making progress more quickly. If we knew more about what's going on with biogenesis and
mm,
the state of cells that produce vesicles that have the characteristics that are desired for a particular outcome, that would be really, really help move the field forward. So we're working on this issue. I'm actually working with some folks at the FDA on this,
mm,
and trying to adapt some of the knowledge that they've learned from trying to develop standardization and quality control for cell therapies, and see like how much of that is relevant and can be useful for EV based approaches. But that's just one of many things, and
sure.
yeah, there's a long way to go, but it's certainly very exciting, and you see a lot of companies and getting into the mix now, and so that'll help drive things forward as well.
Yeah, absolutely. I think there's definitely a lot of different aspects of the problem that need to be tackled. So, I think each one of us can certainly contribute in different areas. And I hope that together as a community, we'll all grow and get to the next level. So, yes, I absolutely agree with all of the comments that you've made. So, thank you so much, Steve, for sharing your thoughts and sharing your time with us today. It's been great chatting with you. So, really appreciate your time.
No, I appreciate it, and it was great to talk about these things.
Awesome. Awesome. Thank you.
All right, take care.